Hi, this is Kavita Kundra and welcome to Mscdfsm Biochemistry class. Today we are completing the chapter Vitamin. So, fat soluble vitamins are vitamin A. Vitamin A in 1906 to 1912, Hopkins studies the rat that are fed on diet of casein, starch, sugar, inorganic salt failed to grow and died. It was observed that rat could have survived with a little of milk if added to the diet. An accessory food factor was found. In 1913, two group of workers isolated the factor from butter, egg yolk, cod liver oil and reported that this factor was fat soluble. In 1915, McCollum and Davis named it as fat soluble A. 1920, Rosenheim and Drummond indicated the relationship of vitamin A with carotene. In 1957, More proved that carotene was precursor of vitamin A. Vitamin A is used for those retinoids that exhibit the biological activity of retinol. And retinol is used to describe related molecules like retinol, retinol, retinoic acid, retinyl palmitate, etc. Vitamin A is collective name of the group of fat soluble vitamin. There are two compounds comprising of vitamin A activities: vitamin A1 retinol and vitamin A2 3-hydroretinol. Retinol is usable form of vitamin A and it is also called preformed vitamin A. Structure of retinol. It contains hydrocarbon chain with beta ion ring at one end and an alcohol group at another. It is pale yellow crystalline solid. The hydroretinol or vitamin A2 that occurs in liver of some fish, it has only half the biological activity of retinol. It is different from retinol by possessing an additional double bond between 3 and 4 carbon of beta ion ring. Retinol is found in animal sources and it is the precursor of two forms, retinol and retinoic acid. The alcohol group OH group oxidize in the body to form aldehyde retinol or retinoic acid. Retinol is used for vision and retinoic acid is helpful in transcription of number of genes. The retinol stores in the body as retinyl ester and most common form is retinyl palmitate. Vitamin is doesn't occur in plants, but plant contain carotenoids like beta carotene that get converted into vitamin A in the intestine and other tissues. Beta carotenoids are family of dark pigments called provitamin A. Carotenoids. Beta carotenoid splits into two in the body and form two molecules of retinol and both half are similar to the structure of retinol. Other carotenoids such as carotenoids and gamma carotenoids on cleavage yield one molecule of retinol. Physiochemical properties of vitamin A are: it is pale yellow, almost colorless liquid. It is soluble in fat and not in water. It can stand 100 degrees cooking temperature in the absence of oxygen. Fat containing retinol become rancid and oxidation and destroys vitamin. Retinol also get destroyed by sunlight and oxidation can be prevented by adding vitamin E. Vitamin D. Hugs Bennett recognized the nutritional value of cod liver oil. 1918 it was used by Tressio in the treatments of ricket. In 1819 Mellon observed that the rickets was responding to fat soluble vitamin present in cod liver oil. In 1931 it was synthesized. Vitamin D has a group of sterols, argocalciferol vitamin T2 and cholecalciferol or activated 7-dehydrocholesterol or also called vitamin T3. Cholecalciferol is a type of steroid hormone made in the skin from cholesterol when the skin is in sunlight. Process of creating vitamin D in sunlight. UV rays cleaves the beta ring of both the compounds ergosterol and dehydrocholesterol and forms ergocalciferol in plants and cholecalciferol in animal in the skin. Dehydrocholesterol converts cholecalciferol in following UV rays. The liver and the other tissue metabolize these vitamin D to 25 dehydroxy vitamin and it further metabolizes into 125 dihydroxyvitamin D3 mainly in the kidney. 1,25 dihydroxyvitamin D3 is responsible for all biological actions. Physiochemical properties of vitamin D. Both vitamin D2 and D3 are soluble in fats and fat solvents. They are not destroyed in presence of acid or alkali. Both compounds can withstand cooking temperature of 100 degrees. Vitamin D is more stable than Vitamin E, also known as tocopherol. Vitamin E is fat-soluble vitamin and function as lipid-soluble antioxidant. In 1923, Evans and Bishop revealed its presence. In 1936, Evans and co-worker isolated it from wheat germ oil and also named as tocopherol. In 1938, synthesis of vitamin was accomplished by two groups, and vitamin E is a group of compound called tocopherols, which is the derivative of tocol. Four tocopherols like alpha, beta, gamma tocopherol have been isolated and they differ from each other in the number and position of methyl group attached to aromatic nucleus. Tocopherols have two methyl group attached to the aromatic nucleus and alpha tocopherol have 3 and 1 methyl group respectively. Alpha tocopherol is commercially synthesized as potent. Structure of vitamin A. Tocopherol consists of ring portion called chromane head and a ring and tail portion called phytyl group. And chromane head has two rings which are essentially naphthalene with one carbon atom substituted with oxygen. Thus a cyclic ether and phytyl group consist of saturated 16 isoprenoid. Isoprene is a branch chain unsaturated hydrocarbon of 5 carbon atoms. Tocotrienol are essentially identical to tocopherol except that they have 3 double bond in the tail at 3, 7 and 11. Properties. They are yellow oily liquid. They are soluble in fat solvents. They can withstand above 100 degrees. Vitamin E is destroyed in the presence of oxidizing agent. In alkaline medium, tocopherol are destroyed and they are stable in acidic medium. Ultraviolet light destroys vitamin. All these compounds exhibit antioxidant property. Vitamin K. Vitamin K is important for blood clotting. In 1934, it was observed by Dam and Skonder that bleeding was unrelated to vitamin C deficiency, could be prevented by alpha-alpha or decayed fish meal. In 1935, it was extracted by Dam and named as vitamin K. In 1939, Dam Carrer isolated pure vitamin K1 and in 1939, Daisy and co-worker isolated vitamin K2. Several compounds having vitamin K activity is known. They are the derivatives of naphthoquinone. Example, vitamin K1, vitamin K2, vitamin K3. Vitamin K1 occurs in plant food, vitamin K2 synthesized by bacteria, and vitamin K3 is a synthetic compound out of all menadione. That is, vitamin K3 is the most potent of all. Vitamin K1 is yellow oil, vitamin K2 is yellow solid, and they are soluble in fat. They are sensitive to alkali, light, and stable to heat.